with the University of Washington eScience Institute and also in civil and environmental engineering. I'm very happy to coordinate with Dave, the University of Washington Data Science Seminar. And we have today Wu-Jung as our first speaker in the series. Wu-Jung is a senior oceanographer in applied physics labs. She is interested in sound, right?
Yes, exactly.
and mostly how to use sound in various media like water or tissue, like animals, right? And with how can we make sense with the data that we collect in the oceans, for example? And so I think Wu-Jung is interesting in answering two big questions that can help advance her research and research in general in this field, like how to sample from an ocean of data, right? And then how to infer something useful from, again, the samples that we collect. I would like to invite Wu-Jung now to start her presentation, and I'm looking forward to watching you.
Okay. Great. Thanks everybody for being here. This is certainly my first in-person talk in the last 18 months or so. Thank you for the introduction. Today, I'm going to talk about our work basically in developing data-driven method and also software tools to study marine ecology using large echosounder data set. I think maybe I should change it to this. All right, that's fine. I'll just keep it as a cursor. Before I start, I want to first acknowledge my collaborator, if I can change. Yeah, all right, here we go. So I want to first acknowledge my collaborators, Valentina Staneva, who's at eScience Institute, and also Emilio Marioga at APL, also the Applied Physics Lab. I'm just gonna be moving this bar back and forth depending on what it blocks, right? So the four people, Don, Daria, Linda, Imran, who are working with us in the Echospace research group. In Echospace, we look at ocean acoustics and data science problems, and occasionally look at acoustics problems also on land as well. All right, so I want to first put my work in the context of active acoustic sensing. So in active acoustic sensing, basically we send out sound and then analyze the echoes to observe and understand the environment. So what you see on the left-hand side here is a typical scenario where you have a human-made sonar system mounted on the ship and trying to observe the interior of the ocean using sound. On the right-hand side are echolocating animals like bats and dolphins, who also use their biological sonar system to forage, like to catch prey and also to navigate, right? So the human-made sonar systems are particularly effective in collecting synoptic information over different scales, like different temporal and spatial scales, whereas the echolocating animals are really good at rapid identification and tracking at short ranges, because a lot of times they want to catch that thing they are tracking, right? They want to eat it, basically. So my work actually spans both of the human-made sonar engineering side and also animal echolocation. In this talk, I'm going to focus on the left-hand side, the human-made sonar type of data application in the ocean. So here we go. All right. So sonar systems are the workhorse for studying marine ecosystems or like mid-trophic level animals in the ocean. What I'm showing here, you are going to hear me saying sonar systems and echosounders somewhat interchangeably in this talk. Echosounders, some of you might have heard of, or fish finders that people use on the boat, are high frequency sonar system that has been spatialized, sort of made spatialized to observe fish and zooplankton, right? So this is a typical scenario where you have a boat and you have a downward looking sonar system and trying to basically look at what's in the ocean. And in this particular case, I just depict a whole bunch of fish, right? So if you have the boat driving along a particular transit line, and then you basically send your sonar pins. And then as you collect the echoes back, you just line them up along with where you are driving by, right? And if you map the amplitude of the echoes, low to high to blue to red, you end up with this figure. The vertical axis is depth, the horizontal axis is depth, is distance or time, right? This is what we call echogram. So when you look at ocean sonar data, a lot of times you're looking at echograms. So what you do is you look at this blob and you are trying to make sense out of this blob, right? So a lot of times what we do is to figure out how do we interpret the echoes for biological information. And in this particular case, it can be, how many fish there are, what are the species, what's the distribution, what's the variability of all these different parameters. So those are the type of information we're after when trying to use ocean sonar to study marine ecology. Yes.
Actually hide that bar.
Oh, really? Great. Yes. Oh, great. Awesome. Thank you. I couldn't figure out which hide that was. Fantastic. All right, cool. So now maybe I should hide this. All right, this is okay. Let me know if this blocks any critical information. All right, so now we know what sonar system can do. I want to show you two examples of how the sonar system see animals underwater, right? So on the left-hand side is a scenario where it's similar to what you have seen earlier, right? So we actually have a downward-looking echosounder mounted on the ship and just drive through a particular survey area. And in this particular case, I label the echogram, right? So you can see the vertical axis, again, depth. The horizontal axis is time, but it's also distance because the boat is a moving platform, right? So there's a confounded time and space axis. And you can see that using different frequencies of sound, you can actually map the distribution of different type of animals, in this case, over the continental shelf. Right? So this is one example. On the right-hand side, I want to show you another example. It's different. So instead of having the sonar system mounted on the boat and looking downward, in this particular case, you have the sonar system in a fixed platform, like a moored platform in the ocean, and actually looking sideways. And that's what we call the horizontal looking sonar system. In this case, we typically use a lower frequency one. So this one is a mid-frequency range around 3 kilohertz. So the axes now are different, right? You are actually getting a bird's eye view of the ocean. Imagine you are like on the sky, you're looking down, right? So the axis here, the X and Y axis, both are distance. And in this case, this is the 3 by 3 kilometer square of the ocean you're looking at. And I'm going to play a movie. And I thought this is cool because in our experiment, we show basically that horizontal looking sonar system can show you how the fish move. In this particular case, reef fish migrating outward from this location. These are artificial reef that were shipwrecks. And around dusk time, in about a span of an hour, you can see the fish basically move out from the reef, right? And it's actually really dramatic. And we observe repeated movement outward. And then during sunrise time, they will move back. And they just do this every day. So it's actually pretty cool that sonar system can do this, right? Yeah, so having seen some examples, I want to talk about this current time we're in. I call it this era of acoustic data deluge, because we have become very good at collecting data, right? In the past decade or so, we are integrating sonar systems or echosounder systems onto all kinds of different ocean observing platforms. So here I'm showing you a few examples. We have the echosounder on the ship. That's a traditional sampling platform. And we have echosounders on autonomous vehicles, both those that travel on the surface, like this one is a wave glider, this one is a sail drone, right? And then the other two are underwater autonomous vehicles and underwater gliders. And on the right-hand side, we also put echosounders on what we call moorings. They are just fixed in the ocean. And these are used both in the Ocean Observatories Initiative, or OOI in the US. Some of you might have heard of it. That's actually in our region. And then also the Ocean Network Canada. So this type of observations actually gave us almost continuous data stream from many, many different corners of the ocean, right? So that's given us this sort of unprecedented opportunities to study marine ecosystems at a scale that's much larger than before, both in time and space. But along with this sort of opportunities, there are challenges, right? So here I'm doing a sort of side-by-side comparison of, oh, I can actually move it off the screen. That's great. Okay, side-by-side comparison of how the data looks like before and now, right? So in the old times, we usually collect data from specific sets of instruments, and that are very discrete. And we usually collect data during dedicated research cruises with a time span to around days or weeks, right? So we know what we're looking for. We go out on the boat, collect the data we're looking for to answer a specific scientific question. But what we have now is a different type of scenario. I have shown you earlier that we are putting sonar system on all kinds of ocean observing platforms. So right now what we have are continuous data flow that have a time span of months and years from all kinds of different type of platforms and instruments, right? So it's a different type of scenario that we need to deal with. So the challenges in this particular case would be, there are a lot of them, right? So here I list 4 of them. The first one is the significantly increased data volume. I don't need to explain this, right? The second one is the heterogeneous instrument sources. And I will explain a little bit about the challenges this creates later on. The third one is that because a lot of these platforms are autonomous, this means that we are actually using acoustics as a remote sensing method. We don't actually know what the biological ground truth is, right? So that can be very challenging trying to interpret in the data. And then the last one is also very critical, is that with all this continuous data flow, we don't exactly know where and what to look for in the data, right? So in this talk, I'm going to discuss our effort in creating, like our effort in addressing a subset of these challenges. One is to create a cloud-based workflow with standardized data to address the first 2 challenges. And then also trying to develop data-driven methodology to find and track patterns in the echogram in order to find where and what to look for in the data. I will start with the second one first. So we're going to look at how we can find where and what to look for in the data by matrix decomposition techniques, right? So before I go into the details, I want to first talk about the type of information we're looking for in the data, right? Because we have sort of gone through a few different things now, right? So the majority of the existing method for analyzing echosounder data has been very much focused on figuring out what the taxonomic identity of the observed animals are, right? So I have previously shown you, like, in these 2 echograms that you have seen before, you are trying to figure out, oh, this is fish, this is zooplankton, and people develop physics-based acoustic scattering models, trying to make sense out of what type of acoustic frequency can help you observe what type of animals and what's the optimal sort of combination for that. And this type of information is very useful in specific contexts. For example, if you are doing fishery survey, you do want to know what you are looking at because you want to count the number of fish that may be there in the ocean, right, of a specific species. However, what I wanted to say is that the patterns that you see on the echogram is also very important in addition to the taxonomic identity of the animals. And to give an example of what I mean by patterns, right? If I don't tell you anything about how fish and zooplankton scatter sound, and you simply look at these 2 echograms, you are probably going to tell me that there are some blobs that look orange and they are associated with different part of the water column. And some of those animals potential animals lay near the sea floor, right? Those are the patterns or the spatial temporal structures in the echogram that we as humans attend to almost immediately when you look at a picture, right? And from my point of view, I think these are the key to leverage large volumes of echosounder data to be able to study marine ecology. To give more concrete examples on what I mean by patterns and what we attend to, here I'm showing you another echogram, right? So in this particular case, again, the vertical axis is depth, the horizontal axis is time. So here you are looking at echosounder data collected by an upward-looking cabled echosounder that is actually off the coast of Oregon. And this is part of the OOI, or Ocean Observatories Initiative, that is installed in our region. And actually, APO and School of Oceanography operates the cable array of the OOI. So in this particular case, right, you can see this is a 14 days of echogram observation. If you look at this immediately, you can see that there is surface wave coming up, right? And then you see this almost sinusoidal patterns across these 14 days of time span. And your eyes just catch this right away. And then you see this bright spot that's sort of in the 50-meter sort of depth that's almost like a layer, right? And I can tell you that this vertical sort of sinusoidal pattern is what we call the DL vertical migration of animals. They basically move up and down the water column every single day during sunset and sunrise in order to forage and to not be eaten during the daytime. And we can go into that if you're interested after the talk, right? But what I wanted to say is our eyes are really good at capturing this type of prominent patterns. But the challenge comes in when we have a lot of data, right? So here I compile a series of echogram and I put it in a rolling format just so that you can look at that. These are 12 months of echograms or echosounder data that was collected at the same site, right? And there are 3 different frequencies of echosounders. So you can actually see that there are a lot of patterns that is really rich and interesting in the data stream. But we can't possibly go through all of it by eye, right? And this is just 12 months. The OOI started operating, the cable array started collecting data at the end of 2014 or 2015. So we actually have like 5-6 years of data that we have to go through. We can use our eyes to do it, but it's gonna take a while, right? So what we wanted to do is to develop efficient and automatic method to be able to extract this type of information out from the echosounder data stream. Right. And so how to do that? We ponder upon this question for some time. And then we thought we can, of course, learn from what others have done using their data set, right? So here we are going to use a matrix decomposition. And here I put matrix and tensor decomposition together because they are sort of the same class of method. But basically, this is a type of unsupervised learning methodology that give you this property of dimensionality reduction. So basically trying to find structures in the data that are simpler than how the data appears. And they are usually more quantitative and more interpretable than the original data set. An example that I'm showing here is what people call tensor faces. So basically they have a whole bunch of face images, and the face images are basically taking pictures of the same sort of people from different angles, different lighting, and so on and so forth. And by using tensor decomposition, you can actually find different axes or dimensions that describe this data. So you can see, like in this particular example, one is different people, the other is different view of the people from different angle. Yes.
Just from a raw dimensionality reduction perspective, are you able to do things like apply like new maps or system plots? Or
yeah, definitely, yeah. So this is just one example. Yeah. So in this particular case, you can find like 3 different dimensions, for example, and then you can use this as a way to just, for in this particular case, you are sending in a query image, and then you can use this type of things, this type of techniques to figure out what the features are in the data. And then actually this particular method spit out what you think this person, who is this person in this particular case. So our view is that we can use the OOI echosounder dataset because it's big and it's pretty reliable and continuous to try to develop methods for data-driven type of method to look at larger time scale and trying to grab patterns out of it, right? So here I'm showing you again a set of 3 different frequency echosounder data stream. Again, the vertical axis is steps, the horizontal axis is time, and now you're looking at 62 days of data in the fall of 2015. And the data, you can see that it actually contains a rich repertoire of daily movement of animals. So previously, I have told you about this DL vertical migration animal go through. If you look closely, you can actually see them basically in the data stream. What we wanted to do is to see if we can use matrix decomposition to actually grab the daily patterns out, right? So what we're interested in doing is to say, if we treat each day of the echogram as a unit, can we then apply the method, like matrix decomposition, different classes of method, to see if we can actually decompose the components, like decompose the echogram into multiple components that when they add up, give you the pattern that you look at across each day. And of course, there are going to be noise and outliers when you add up the components, right? We think that the non-negative matrix factorization decomposition is a nice framework to work with. And there are many reasons to that. But essentially, to introduce this method, I'm having this side-by-side compare the NMF with the PCA, the principal component analysis. I'm sure a lot of you are familiar with. So in the PCA, in this particular example, we're using the human faces again as a way to introduce this, right? So you probably have heard of eigenfaces. Basically, you have like a database of human faces. And when you do PCA on that, you can actually come up with components, which people call eigenfaces. And then you can think of eigenfaces as sort of building blocks, right? Or like components that when you do linear weighted linear combination of the components, basically multiply the components by weight, you can reconstruct one of the pictures in your database, for example. And NMF is a very similar type of method that basically work, the formulation looks very similar. The difference is that for non-negative matrix factorization, we force the formulation to be non-negative. So, you know, the components and the weights have to be both positive. And this type of decomposition gives you this part-based, this property of part-based representation. This is the best view as if you compare the PCA on the left-hand side and the NMF on the right-hand side. You can see that the NMF view, instead of give you eigenfaces that each one look like a tiny face, they actually give you like eyes, eyebrows, mouths, or noses, right? So they are much more part-based, that giving us this view of maybe we can then think about things that are building blocks that add up to be a picture, right? And the other important thing is that because echo energy is additive, so we don't actually do, you know, fish energy minus zooplankton energy when you look at echoes, right? So if you actually work with non-negative reconstruction, it makes much more sense when we're looking at echoes. Yeah, so what we did was basically we organized the Echosounder data set that you have seen earlier into sort of a stack of daily echograms, and then run a specific version of non-negative matrix decomposition, factorization, and we call it tsNMF, right? And what we found was that indeed we can actually extract echogram patterns that are biologically meaningful, right? So what I'm showing you here, each vertical column is one component, and each one component has 3 frequencies. And the 3 frequencies come from, remember the echogram has 3 frequencies, right? So we basically just stack them on top of each other, right? And then run the tsNMF. And then we come up with these patterns, right? And each of these patterns are 24 hours at 200-meter water depth. This is our data dimension, basically. And then along with the patterns, we also find out about activations, right? So on the right-hand side, you can see pattern 1 is corresponding to this line, and pattern 2 is this other line, and then pattern 3 is this other line, right? And what we think is that this actually gives us a good handle on how to represent that very complicated echogram that you saw earlier into a much more compact representation of the temporal processes in the data, right? Because we can basically look at component 1 and then use the frequency features in there and say, oh, look, like this actually does look like the DL vertical migration. And the frequency dependent signature of the echo strength actually look like zooplankton type of animals. And we can do the same thing for component 2 and component 3. And this type of patterns is much more interpretable when you are looking at a much longer time series compared to the original data. Sure, you can look at original data and then describe it. But if we can actually have the activations, then we started to be able to do quantitative analysis. So in our view, this is a very powerful way of thinking about long echosounder time series. And just to show a few examples of the reconstruction, some of these are a little difficult to see in this very compressed form. If you're interested, I can show you, the expanded version afterwards. But in this particular case, you can see, component 1 is this DBM cycle, and we know that it's activated more in this sort of time period. And you can look back on your original data and then find a correspondence in there. The same thing goes with component 2, which is a strong subsurface layer. And you can see the subsurface layer basically dissipated after the first 1/3 of the time period. And you can do the same thing with component 3. But in this case, it's actually really difficult to see component 3. It's much easier to see when it's not there. So component 3 is something in the middle of the water column during the daytime. And you can see that when component 3 is low, the water column echo looks very empty. Basically in this location and in this location. And the other cool thing that we found out from doing this is that because the tsNMF activation gives you a much more sort of like a summarized form of what the temporal processes in the data is. You can then actually use that to describe changes or similarities across the time span you are looking at the data. So what we are having here is a distance matrix. The vertical and horizontal axes are both time. And these are basically just Euclidean distance of the activation. And the red ones means that they are more similar. The blue ones means they are less similar. So you can actually start to see, oh, you can see blocks of days along that 62-day time period that we're looking at. Some of the days are similar to each other, and some are actually not similar to others, right? And then we ran the hierarchical clustering, and then actually you can cut the dendrogram and actually come up with clusters of which groups of days are more similar to other group of days. And what we found really interesting is that we actually, these are the top two panels are only from the echosounder data. And the bottom panel are actually from another type of instrument that's called acoustic Doppler current profiler, or ADCP. You can ignore how it's called, but basically it's a way to measure ocean current directions using acoustic signal, again, using sound. And basically using the ADCP observation, we come up with this bottom panel. These are the north, south, and east, west current directions at the same location. And you can see that the top two panels and the bottom panel came from completely different data sources. But you can see that when we found changes in the clusters from our tsNMF distance matrix analysis, there are actually corresponding changes either in the direction or the magnitude of where the ocean current was flowing. And this is interesting because this means that, I mean, intuitively, the animals live in the ocean. So when the environment changes, the animal will respond behavioral in a certain way, right? But what we think this shows is that our analyses are able to grab the type of changes that can actually enable us to analyze the echosounder data with other environmental data from the ocean in a much more compact representation instead of looking at an echogram in a complicated form as is raw data show. So this basically wraps up what I wanted to talk about in terms of where and what to look. Challenge of analyzing large ocean data set. The next part of the talk, I want to discuss what we are doing in terms of creating a software tool to enable cloud-based workflow and using standardized data. And the sequence is a little bit reversed, but I thought it's interesting to first look at what type of science you can do, and then look at what tools you need, right? And what we're doing in our group is actually to do this both at the same time. And it comes with its own pros and cons, but it's actually very interesting. So now we're looking at what we can do, our effort in creating a cloud-based workflow with standardized data. So here I'm listing 4 distinct type of challenges when we wanted to do integrative analysis of ocean sonar data. Right. So previously, I didn't talk about how I got all those data together, right? But this is the how part, right? So there are a lot of challenges currently. If you have a whole bunch of data from, let's say, different type of ocean observing systems, you want to combine them together. It's actually a very tedious and labor-intensive process for the following reasons, right? The first one is interoperability. There are a lot of different sonar manufacturers. Each of the manufacturers probably produce a lot of different type of sonar models. And a lot of manufacturers have their own software systems. It's proprietary to read and parse and plot the data. And there are also other vendors who just produce software systems to work with data from different systems. But a lot of these are proprietary. Or there would be open source code, but it's written in proprietary programming languages. So there is an access problem in this particular situation. There is also the scalability problem because the majority of the existing echosounder data analysis methods, a lot of those are actually very labor intensive. It requires human expert to actually circle important features in the echogram, right? And also because the data is growing larger and larger. If we want to do interactive type of data exploration, it's typically a slow process. A lot of times people sit there and wait for the data to load, right? So that actually make it a much more tedious and labor-intensive process than we want it to be. And the other is a reproducibility problem, because a lot of the software are actually GUI based. And if you have used a GUI, you must have used a GUI, right? You know how a lot of times you click this first and then you click the other things. You need to remember what you have done. And sometimes some software give you a log file that's very useful, but a lot of times you may not be able to recreate what you have done if you don't remember, right? So it's a lot easier if you actually have a script or something that can be automated to basically reduce your burden in this type of process. And the last, I put community here because if we were to tackle all of these challenges, ultimately we are talking about actually transform the workflow of working with Echosounder data. And along with this, there is an embedded challenge there is that with all of different sonar models and manufacturers with their own data formats, we need a convention for us to be able to actually pass the data between different types of instruments so that we can just work with the data instead of struggling with the files, right? So our approach to do this is to create, our goal is to eventually build an interoperable, scalable, and reproducible pipeline. Right now we are building a software that's called echopype. The goal is to streamline the tedious data workflow by leveraging tools that's already existing in the scientific Python ecosystem. So what I'm depicting here on the left, from the left to the right-hand side is a typical Echosounder data analysis workflow, right? You have the raw data, you do some pre-processing of the data, do quality assurance, quality control, calibrate your data, and then you wanted to go to visualize and analyze the data. And eventually you extract information from the data that goes into ecological or fisheries interpretation or analysis, right? And so what we are aiming for with echopype is to use the libraries that we that I grabbed their logos from to basically use existing capabilities so that we can connect the data workflow to the cloud and being able to basically leverage what's already there to accelerate the processing of Echosounder data. This is the echopype workflow. Basically what we're looking to do is we start with a whole bunch of very heterogeneous or very diverse type of data formats. We convert the data or we standardize the data into a format that is called the NetCDF data model. Some of you probably have seen this data model before or have heard of it. Basically, it's a type of data that is a type of model that, or the format that is self-describing because it contains metadata. For example, if I give you a numerical array, but I don't tell you anything about that, you don't really know what I'm plotting, right? But if you get a NetCDF file, what you get is actually the data that labeled with axes. For example, the Echosounder data will be labeled with like time and depth or time and range from the sonar, right? And this type of data typically is much easier for random access. This means that you can go to a terabyte of file, actually just get a small slice out of it instead of having to load the entire data into memory, right? And for NetCDF data, in echopype, we are following this convention that is called the Sonar NetCDF4 convention version 1.0. This is the first convention for raw Echosounder data that was just developed like 3 years ago. So we basically standardize, in our workflow, we standardize all the data from its raw format into the same standardized NetCDF data model. And from there, we do calibration analysis and visualization. Another reason, there are two reasons in addition to the nice thing about NetCDF model itself to choose this approach is that once we standardize the data, we can provide the users with a uniform interface so that as a user or as a scientist, I don't need to know exactly how to analyze different data from different type of instrument differently. Once they are calibrated and come with physical quantities, for example, what is the strength of the echo, then I can just use data from different instruments pretty well. The other is that because NetCDF is a very widely supported format in geoscience and earth science, there are a lot of existing libraries, for example, distributed computing using cloud or cluster resources. Those things are already existing. So if we just leverage the existing capability, these open source libraries, we can actually move faster in a specific domain. And here we are just looking at ocean sonar type of data. Yeah, and it'll be nice if I can actually do a demo, but you know how when you try to demo something, it may not run. So I just take screenshots, right? So what I want, and you are all welcome to go up to this repository where we actually have a binder link that you can evaluate this particular demo notebook by yourself if you want. But what we are doing here is to demonstrate how you use echopype to pull the data directly from the OOI raw data archive, and then combine the Echosounder data with the ocean sonar. The OOI also have the sonar solar radiation data. And what we're doing here is we pull the data from the day of the solar eclipse a few years ago. The OOI, that particular upward looking Echosounder happens to be in the totality zone. So you can actually see very cool pictures, and I'll show you in the next slide, by comparing, basically pulling together Echosounder data stream and the solar radiation data stream. You'll be able to see the animal's response to the solar eclipse. And it's just a very nice sort of one-day type of data that illustrates what we can do because we have echopype. Because honestly, before we have it, you really have to jump through hoops. You probably have to toss data across 2 or 3 different type of software systems to be able to do what we can do. And now we can do everything in one notebook. It doesn't look like a lot, but it actually took a lot of steps to get there, right? So what I wanted to show, just very short code snippets, right? We can basically pull the data and calibrate it and then save it into a file for visualization using 4 lines of code, right? And so all the work has been hidden underneath. And that's what we want it to be, right? We want it to be a uniform, easy to use user interface so that we can combine different type of sonar data together. So this is a picture if you evaluate that notebook by yourself. On the top panel are the solar radiation that's measured at pretty much near in a very nearby location compared to Echosounder on the surface. You can see there is a dip in the solar radiation. This is a time of the solar eclipse. And remember, I previously tell you about the DL vertical migration of the animals, right? So the animal typically try to avoid being eaten by visual predators in the daytime. So they will actually dive to deeper ocean. Most of the animals will try to dive to deeper ocean during the daytime. So this is daytime, right? And they dive down. But as the sun gets covered by the solar eclipse, like by the moon, basically, the animal gets tricked and actually move upward until the moon moves away and then went back down again. So this is actually pretty cool. And the fact that we have the ocean observatory that's already there allow us to observe this. So to me, this is a very interesting case where you have the capability that's already existing as an infrastructure, but you are able to make discovery because it's already there. It's very different than we know the solar eclipse is going to happen in this time, so we charter a research boat so we can go at that time to do that sample. It's a very different scheme, and I found it very interesting. And another exciting thing is that we started this because I want to analyze the Echosounder data from the OOI, but now we have gone full circle again. Like the OOI data team actually now use echopype to produce data products to serve users. So we're pretty happy with it. And of course, we're still developing, but this is a nice sort of collaborative situation right now. So just to wrap up, going forward, there are quite a few things we are doing right now. I only show you a small snippet of the OOI data and the analysis method we apply to it, right? So right now we wanted to basically expand the spatial and temporal coverage of the analysis. What I showed you earlier are data from this red triangle, but in fact, the Ocean Observatory initiative have 6 different sites that is along the coast of Washington, Oregon, and they are literally streaming data all the time. And they have been in operation for like 5 to 6 years now. So we have actually a lot of data to work with. And this is a small screen grab of what the OOI has in their endurance array. And you can see anything that was the sort of yellow cone are acoustic instruments, because acoustic instrument or sonar systems are actually very widely used for ocean observation. And I'm just showing you the instance of the example of using those to observe biology. You can use those for observing currents or many other things, right? And on the right-hand side is we are also collaborating with the NOAA Fisheries, the Northwest Fisheries Science Center for their fishery survey. So here you can see their data came from the entire west coast of the US, from the southern part of California, all the way up to the northern end of Victoria, the Vancouver Island in Canada, right? So this two sets of data actually give us a very nice spatial and temporal span on the west coast of the US that we can work with. So we're really excited to like start working on this right now. The other thing we are doing is to try to connect the sonar observation with ocean environmental variables using similar dimensionality reduction type of techniques. Because the ultimate goal is to figure out why the animals are doing what they're doing. Is it because of something of the environment changes? And we all know the implication of this in the context of like the climate change situation that we're in right now. And of course, we continue to grow echopype. And in part of the process, we wanted to broaden the engagement with the community, both for the more fisheries acoustics or ocean acoustics type of community, and try to connect also with the oceanography community as well. So I think I will just start here. I'm happy to take questions. We are hiring, so if you're interested in doing research with us or software development opportunities, let us know. Thank you.
All right, I can't tell you how awesome it is to clap in person for somebody to give me. Questions for our speaker? I have one from Zoom.
Okay.
That I'm not going to try to read because it has code in it.
It has code in it?
Yeah.
Did it arrow out? Sorry, I have to
There
ask.
you go, sorry. And if you could read it so that
How does echopype calibrate the EKA spin? Oh, how does it calibrate it? Oh, interesting. Should I answer live?
yeah, sure.
So I just answer it?
Yeah, go for it.
Oh, the calibration, it uses, I know Sam, so I assume that I'm trying to answer the question that he's asking, but feel free to tell me that's not a question you're asking. We're just pulling, the default is to pull the calibration coefficients from the data files that basically store those coefficients, but you have the options to actually pass in the environmental coefficient or environmental variables like salinity, pressure, and depth, and stuff like that, temperature, to calculate the absorption or sound speed. Those are the environmental parameters that's needed to calibrate the measurement from Echosounder. Okay. Yes.
I have a data format question. Have you heard of HDF files?
Yeah.
What is the difference between those and the ones that you're using?
So my understanding is the NetCDF.
Can you repeat
Oh.
the question? Just repeat the question, so.
Oh, yeah, thank you.
I can
Yeah.
hear it, yeah.
If the question, if I understand correctly, was the HDF5 data format. How is that related to NetCDF? My understanding is that the NetCDF data model has gone through a few sort of backend support. So HDF is used in one of the versions, one of the recent versions. And I think they are also building additional backend right now that's more cloud compatible.
Can I ask a question?
Yeah, please.
I'm just curious about how you interpret the data. Like we looked at the methods, but for example, did you look at cloudy days versus clear days? Or can you say something about how many fish there are? Or like if they're changing their patterns of migration, like by using the sequence of days or years? Because if
yeah, because there are so much data.
the conditions are changing, then the patterns will change as well.
So
The same skills support,
I know that's like, yeah. I guess my honest answer is that's what we are trying to do, right? Because there are all these hoops to jump through. But it is true that we know, or I guess like other people have found that, for example, the DVM behavior does change depending on the type of days or the weather conditions. And those are the things that we actually do want to pull out from the data. Because one of the things is that if you can isolate the DVM patterns, it just gives you a nice read of, for example, what's the depth of the DVM. And that's something that a lot of people have worked on. And we are really curious about, what is that behavior in the OOI array we are looking at. Yeah. Okay. Do you want to choose? Okay. I'll just go with the front person. Yeah.
So during your multiple parameters during your dimensionality reduction process, like, do you have when you and your team are like like reducing the dimensionality to like try a lot of different methods, and like what's like the standard that
Okay, I'll try to repeat the question to see if I get it. So your question was, how do I choose which dimensionality reduction method that I was using and the parameters of that? Yeah, so we went through a few different methods, and when I started, I literally just try to find existing libraries that I can apply to the data. And then eventually the one that we used was actually an algorithm that someone proposed and was written in MATLAB. So Valentina actually basically translated and sort of coded into Python, and then we use that for our analysis. The major challenge in that process for us was that that particular formulation has a few parameters that we can tune. And tuning parameters, I'm sure a lot of you know, like you can tune many things, right? So, and you will change the result and what you are getting, right? In the process, what we did was we looked for the most stable output. So we can tune different parameters, and we found out some of the parameters is basically sensitivity analysis, right? Like we know some of the parameters are not really sensitive in terms of producing output. And the other is because we are looking for temporal smoothness in our decomposition so that we can actually look at a longer time span of data. We do manipulate that particular parameter. And there is a trade-off between how smooth your decomposition is and how much sort of noise or residual that you throw away. So we look for a pattern that basically is a sweet spot between making it smooth enough, but also not producing too much reconstruction error with the original data. And that's how we select those parameters.
So it's like essentially like an optimization between smoothness and noise, right?
Yeah. Yes. Yeah. Yes.
When you're going through the decomposition process, you said you had 3 different variables or 3 different processes that were weighted against, and you said that one of them or 2 of them were zooplankton-like and one of them was fish-like.
Yeah.
Have you all gone through and with the observations like at the site to demonstrate that that's actually going on with the fish or that's actually going on with the zooplankton? Like, how are you able to assign like those functionalities to those characters?
Right. So the question was, in the interpretation, I said that there were 2 patterns that looks more like zooplankton and one looked more like fish. How did I know? I didn't, and I don't still, because that's one of the things that, as I said earlier, one of the challenges which I didn't address in this talk was that we don't have a lot of biological ground truth data to be able to verify acoustics. So that is actually one of the challenge, or I think something that, as a community, we should improve on, right? Because the OOI observing platforms or infrastructures are there, but a lot of information you do have to be there or have some sort of in situ positive identification to be able to know what it is. For example, people have used net toes or cameras to figure out what you're actually looking at in reality. During that particular time span, we unfortunately didn't have biological ground truths to compare. But the reason why we can infer potentially those may be zooplankton or fish was because we do have the knowledge of how different sizes of animals scatter sound differently. So by comparing the echo strength at different frequencies, we can infer this is more likely to be zooplankton type of echo or more likely to be fish type of echo, but we don't know for sure.
Okay.
Yeah.
And then, just as a quick follow-up, is as part of the pipeline development, is there going to be more integrations with that biological ground truth data or developing like collaborations or other research projects with groups that can provide that type of ground truthing?
Yes, for sure. Yes, yeah. In fact, we are trying to, we are trying to get a proposal in actually basically to do that, right? Because the workflow right now is the acoustic workflow. And as you said, there is a biological workflow that should be integrated in. And it's actually a very well-worked path, right? Like many people have done that integration. But one of the key things is there is, again, an embedded data standardization problem in there. It may not be difficult in itself, but if everyone comes up with different spreadsheets, then you have a problem, right? So, yeah.
Thanks.
All right, I hate to cut it short. I think this just reflects how great of a talk it was, and so let's all thank our speaker again. Thank you all for being here in person. I really appreciate it. And what a wonderful way to start this off. This is just great. Thank you so much.
Yeah, thank you. This was fun. And it's a little like, I'm not sure where to look, but it's all
You
right.
did great, because I don't know if you saw, and actually, let me, yeah, I'll stop the, oh, wait, we got another, oh, Tucker asked the question. Oh, let me stop the, no, this is okay.